So my group recently is interested in the weak I signaling in cytosolic DNA synthesis pathway. And today I will show you like a critical component in DNA synthesis pathway. The STIN is regulated by weak I signaling. So weak I or weak I-like receptor is a cytosolic RNA receptor. This they play quite an important role in synthesizing RNA virus or like retrovirus can trigger like innate immune response. Unlike the TLR, the expression of TLR is cell type specific, but like weak I can be detected in almost all the cell lines. So they have three members of weak I, weak I-like receptor. It include weak I, MDA5, LGP2, and they also have two adaptorprotein. One important one is MAVS, mitochondrial adaptorprotein, and also a STIN, also like have play some role in the weak I signaling pathway. And in these three RLR members, only weak I in MDA5 can detect induced interferon antiviral response, not LGP2, because the trigger the downstream signaling pathway require the N-terminal cardobin, and the LGP2 did not contain the cardobin. Well, both weak I and MDA5 can recognize RNA in trigger. Recognize RNA in trigger the signaling pathway, but the function of these two receptors is not redundant. MDA5 is mainly can recognize long double-strand RNA, and also like the MDA5 mediated signaling need the cooperation of the LGP2. LGP2 can facilitate the MDA5 binding to RNA and trigger the signaling pathway. While the weak I, they can recognize the short double-strand RNA or 5' triphosphate RNA. In the binding of RNA is through the C-terminal, like C-terminal domain. As shown here, just show like the C-terminal domain of weak I can bind into the RNA molecule. As I mentioned before, in most cell types, weak I is expressed in most cell types, but the weak I alone is also can be induced by interferon. So in unstimulated cell, the weak I exists as a latent form. The binding of virus RNA, they can induce polyexplanation in phosphorylation in the C-terminal, in the N-terminal cardobin, and this will lead into the conformation change in open the cardobin, and the cardobin can interact with mitochondrial adaptorprotein, MAVS, and this will induce oligomerization of MAVS protein and activate the MAVS. In the activated MAVS, will recruit downstream single molecule such as 12-6 in LIP1 NEMO. And this can activate the canonical IKK kinase and activate NF-kB and induce inflammatory cytokine. Well, when the activate MAVS can also recruit 12-3 NEMO and tank, and this will activate non-clinical kinase, the TBK1 and IKK epsilon, and this will activate IRF3 and IF7 and induce type 1 interferon production. In the release of type 1 interferon can bind into cell surface type 1 interferon receptor. And this will trigger the JAK start signaling pathway and activate a larger number of interferon stimulator genes. And some of these IgGs have intrinsic antivirus activity, such as mixed protein, ADA, APOBEC3 in TRIM5 alpha, IFITM, LG15 in Vaprin. And some of the IgGs can inhibit the virus through indirect mechanism, that's through the inhibition of cellular transcription, such as to have a INSL system. Also, some of the IgG can inhibit cellular translation, such as PKR, FG56 in FG20, and this will lead into the inhibitor of virus replication. And recently, we have used like 5' PPP RNA as to trigger the weak eye. And we find like the administration of the 5' PPP RNA in the mice can protect against the lethal challenge of the influenza virus. And also like in cell model, like treat the cell with 5' PPP RNA can protect against several different viruses. These include influenza virus, dengue virus, hepatitis C virus, and also like Vaccination virus. So to study the mechanism of the 5' PPP RNA action or the weak eye signaling, we perform a microarray to compare the genes induced by 5' PPP RNA in interferon. And we can see much larger number of the genes can be induced by 5' PPP. And the interferon-induced protein is all of the interferon-induced protein is can be induced by 5' PPP RNA. And as effect, like the 5' PPP induced gene can convert to the activator of IRF involved in the interferon signaling. But some of the immune-regulated genes can be also regulated by, quite strongly regulated by 5' PPP RNA. These include the genes involved in the communication between the innate and adaptive immunity, and also the protein involved in dendritic cell maturation, and also like antigen presentation pathway. And as I mentioned before, the STING also is an adaptorprotein in weak I-synchronous pathway. But much more important role of STING is in involved in DNA cytosolic DNA synthesis pathway. The recently study indicated like the STING can direct bind into the cGAMP, the cyclic GMP-AMP. And the cGAMP is synthesized by the cGAS, and the cGAS is activated by cytosolic DNA, and upon activated by the DNA, the cGAS can utilize ATP or GTP to synthesize the cGAMP. And also STING also is combining bacteria second message like cyclic AMP or cyclic GMP. In several other cytosolic DNA sensors, such as IRF3 dye or DDX41, may also use STING to trigger the downstream signaling pathway. And the activated STING can, like weak I-synchronous pathway, they can activate the TBK1 in activated interferon, type 1 interferon signaling. Also, like STING can also activate the STAT6 in induced STAT6 dependent response. In our recent study also indicated like STING involved like in can trigger like apoptosis in human T cell leukemia infected cells. Our study like indicated like the HTLV infection of monocyte can lead into the produce like transcription intermediate reverse transcription intermediate DNA. In this RTI can activate like the type 1 interferon or activate the apoptosis through induced Bax protein. So we are interested like in how STING protein is being regulated. So my student Grace like performed a study like in A5 4-line cell use Sendai virus infection. In using the Western blot, we can detect like the IgG protein being activated by Sendai virus, and she also detect like STING is quite strongly induced by Sendai virus. And the induction of the STING is in the iron level because here like use qPCR can detect like STING is quite strongly induced by the following Sendai virus induction. And the induce of STING expression by Sendai virus is not limited in A5 4-line cell. In other cell types, such as liver cell, HU7, in prostate cancer cell line, PC3, and also like in U87 cell, all these cells like virus infection, RNA virus infection can induce the STING expression. And to determine whether the virus-mediated STING induction is through the weak I pathway. So first we use like a weak I mutant cell, like HU7.5 cell. HU7.5 cell is derived from the HU7. The only difference is this HU7.5 cell carry a mutation in weak I catecholamine. And we can see here in in the weak I deficient cell, the Sendai virus induction did not trigger the IgG gene expression and also did not induce STING expression. And we try to use the 5' ppRNA, and similarly, 5' ppRNA can induce STING expression like in Q7, but not in Q7.5 cell. Also, like in MAVS cells, like STING can be induced by Sendai virus in wild-type MAVS, but not in weak I knockout MAVS cell. And finally, like in we know like MAVS is an important adaptorprotein in weak I syncytin pathway. So in A549 cell, when we use siRNA knock down the MAVS, it will block like virus-mediated activation of the induced of the STING. So then my student performed kinetics to in here he's in the data show like unlike the weak I can be quite rapidly induced by weak I in IG56. These two IgG can be quite rapidly induced by 5' ppRNA, but the STING induction by 5' ppRNA only can be detected after 24 hours. But the induction of STING can be last for 5 days. Also, like STING induction can be detected in vivo, like when we challenge mice with 5' ppRNA, we can detect the STING induction in kidney, liver, also in lung tissues. And we know like weak I can activate the NF-kB in IRF signaling pathway. So we try to determine which transcription factor is involved in the induction of STING. In here we can see knockdown IF3 or knockdown DLA or P65 NF-kB can partially block the virus-mediated induction of STING. While if knock down both factors, you can completely block the STING induction. And because the induction of STING is a little bit late, so we think maybe this is due to the autokine or parakine mechanism. So to test this possibility, first we treat the cell with 5' ppP for 24 hours, and then we halve the supernatant and use supernatant to stimulate or freeze cell, and then we detect like the STING induction after 24 or 40 hours of the supernatant treatment. And as we can see here, the supernatant from 5' ppRNA treated cell can induce STING induction is as nice as the cell treated with 5' ppRNA. And again, this requires like intact interferon signaling, because when we use this supernatant to treat like HEC1B cell, this cell lacking type 1 and type 3 interferon receptor. In this cell, STING cannot be induced either by 5' ppRNA or the supernatant from the 5' ppRNA treat A549 cell. So then we see like use like treat the cell with interferon in TNF. We can see like treatment of interferon in TNF can induce sting expression. And we also check the type 2 and type 3 interferon. And we see like type 2 interferon can also synergistic with TNF to induce sting gene expression, but not type 3 interferon. Interferon gamma did not induce sting. So here just show like the induction of sting requires type 1 interferon receptor. If we use siRNA knockdown type 1 interferon receptor, you will block the sting induction. Also, START1 is important. If you knock down START1, knock out START1, the sting is not induced. So, as conclusion, like our data demonstrate that the Sendai virus infection can induce sting expression at both the RNA and protein level. In sting, induction is dependent on function RIG-I signaling. In 5' PPP, 5' PPP RNA triggers sting expression both in vitro and in vivo in the mouse model. And finally, the TNFα in type 1 or type 2 interferon can synergistic regulate sting expression. So my student Grace is do most of this work, and also thank my collaborator in the funding from the CHR, Cancer Canada, and also from the Quebec government. So thank you.